joined us today to talk about soil health and the priorities to take on improving crop production, as well as how we can assess soil health in low nitrogen fields for potato production.
Okay.
You want an umbrella, Jonathan? You good?
So, I guess, thank you to the organizers for inviting me to come out. For any of you that have done any looking into soil health, the first part of the presentation is probably going to be old hat, but I didn't know exactly who was going to show up, and so I thought we would kind of start with a little overview just to kind of set the stage and make sure everyone's on more or less the same page. So we're going to talk a little bit about why soil health is important, mostly from a plant sort of health perspective, but I will talk a little bit about things on like how you define soil health, which I think really depends on what you're trying to achieve rather than there being a single definition, but I'll get into that. Then we're going to transition into some ways to assess soil health. My research is on potato health, so I come at it again, from the perspective of soil health supporting plant health, but there are other definitions and things that you might want to measure, and I'll talk about that briefly. But I also don't kind of view myself as a soil health expert, except for how it affects plant biology. So I'm not going to say like this is the most important soil health measurement. Rather, I'm going to ask the question, can we change how we measure soil health with an emphasis on, say, like automation, robotics, high throughput data in order to be able to ask different questions? That's kind of a bit more in my wheelhouse. So we're going to talk about some of the protocols and methods we've been developing to try and do that. And then at the end of it, I have a bit of a case study that we've been working on with a local grower to kind of ask this question. So if this is what we want to achieve in terms of an outcome, what can we measure? How can we use that data to decide whether we're going in the right direction? And where do we go from there? And then I'll talk a little bit about what we've going on here, depending on how much time we have left. So I think I have enough for everyone, or you can look on with someone else. I'm just going to get started on the first page there. So soil microbial function, I think, is what's really important in terms of soil health. So again, from a plant biology perspective, it's about what the microbes are doing to support plant growth, or at least that's what I'm interested in. There's lots of functions the microbial community can and does perform, but a few examples I have on here that I think are particularly important are things including nutrient cycling, stimulating plant growth, and then what I call antagonism that's involved in disease suppression. I included a couple pictures, and then at the end there's a reference section in case you want to look up the paper, but the first one on nutrient cycling just kind of gives an example of some of the nutrients that cycle throughout the soils that plants can take advantage of, that microbes are responsible for. So these are things like carbon, nitrogen, phosphorus, and then a bunch of other metabolites. On the soil plant growth promoting perspective, there are a group of bacteria and fungi called plant growth promoting rhizobacteria, or PGPR. And a lot of bacteria will produce chemicals that are similar to plant hormones. And so when these bacteria are present, they're essentially stimulating things like root growth, root elongation. And so having a healthy soil that has a robust, healthy soil microbial community will alter things like plant architecture, which then has an impact on how well those plants are able to take up nutrients like water and then support growth. So that's a really important function for the microbial community. And then I mentioned the antagonism. So has anyone heard of the concept of disease suppressive soils? Maybe, maybe not. So we don't necessarily know exactly what's going on there, but there are some soils that it seems pathogens have a really hard time getting established. And even in some cases when there's monoculture, which you would think would promote disease accumulation. And from what we believe is going on is that it's mostly a microbial community function. And so microbes can do this in a few different ways. They compete with each other in the soil environment. And so some microbes will produce antimicrobial peptides and other chemicals that will suppress the growth of pathogens. And then there's also this concept of pathogens and beneficial microbes competing with each other for space. Verticillium with potato is a really important plant pathogen, but verticillium is actually a genus of a whole bunch of species. And some verticillium are responsible for early die, but a lot of verticillium isn't. And so it's thought that you can have examples where, say, one species of verticillium colonizes the plant, and that actually prevents the pathogen from coming in and colonizing it. So you have all these, the point being is that the microbial community is just really complex. It's responsible for nutrient flow throughout the system. It's responsible for communicating with the plant and with others. Yeah?
And is it possible that these anti-soils, all of a sudden the predatory population of bacteria that's keeping the pathogens at bay collapses?
Yep. Yeah, so dynamics are, and we're going to talk about it in a little bit, but the dynamics are very complex in both space and time. So you can have microbial communities in the same field, but depending on the time of the growing season, the community will change, and so on. What seems to happen with the disease suppressive is that the community becomes stabilized, and it happens to become stabilized in such a manner that pathogens are unable to become established. And what's really interesting about it is, again, like the classic examples, like if you look in the literature, a lot of it's been studied in China with rice paddies. So you have these rice paddies where every year they're just growing the same thing and they're putting like all this nitrogen into that. And like a lot of things that you would consider to be unsoil healthy things to do, like so no crop rotation, lots of chemical fertilizer and all that other kind of stuff. But for some reason, it's become stabilized such that it actually could be classified as a healthy soil. Now, this kind of leads into the next little bit, and I'm glad you asked it, is because the definition of what is a healthy soil versus a not healthy soil, I think largely depends on what you're trying to accomplish. So if you're trying to, grow a cash crop, and your concern is about getting yield and quality out of your crop, so with potatoes, if you're growing for French fries, good French fries, wheat, whatever you're looking for, what you're going to define as this healthy soil is going to be very different than, say, you're trying to participate in a carbon credit market, where your objective is to have carbon accumulation in the soil. Or maybe your objective is we have a real problem with soil erosion. And so that's the thing we're trying to solve. Or we're trying to control for a pathogen, so say scab with potato or anything else. So that's where, a lot of what I think you have to consider with this is that what you're measuring, how you're defining soil health has to be based on your objectives, okay? So getting onto this kind of concept of healthy soils, microbes don't operate in isolation with each other. As we've kind of talked about, it's this really complex interaction between different microbes, the plant hosts, and all that kind of stuff. Generally, higher soil microbial diversity is higher soil health. And there's a few models that can be used or theories that can be used to explain that. And that's what's on page 3 under figure 2 is showing a few examples of that. So there's this idea, what's really important isn't who's present, but what functions are being performed. So again, if you've got pathogens, functions that work against those pathogens, that's going to be important. If you've got high pH, acidifying bacteria are going to have a big impact on making phosphorus and other nutrients available. If you've got low carbon levels, that means there's less energy available for the microbes. So you might want to bump your carbon levels up. So one explanation for why diversity matters is this concept of insurance or redundancy. So again, it doesn't matter who's present as long as the function's being performed. And if you have higher diversity, the chances that someone's able to do the function that's required is going to be higher. There's also the idea of complementarity. So microbes can complement each other in space. So you'll get different microbial communities, say in the top few centimeters of the soil than you will have lower. You also have changes in the community composition over time, and they can complement each other. So think about like if you've got a field that's got a whole lot of plant residue and you want that plant residue to be available to support plant growth, well then decomposers that are eating that cellulose, releasing the nitrogen and all that is going to be really important. And that's going to potentially complement, say, one of these antagonistic pathogens. So they're able to use some of the sugars and the carbon that's released by the decomposers, and then that can be used by the plant itself, but it also feeds the other microbial community. And then you have this idea of synergism. Yep? No, I'm fine. So with synergism, the idea is that you have these microbes that can operate in isolation, but the effectiveness of how much they promote soil health and plant growth is augmented or increased more when they're working together. So you think about like nitrogen fixing bacteria, they can provide some nitrogen into the soil, but if the plant species you're growing aren't able to take up the form of nitrogen they're producing, in order for you to get the best effect, you have to have something that converts it. Okay? Now, these three theories, a lot of them are kind of borrowed from other fields of study. So like the insurance, or when you're like doing investing, the idea of redundancy and synergism and all that, it's kind of adopted from that. And it's not to say that any of these theories are, at odds or mutually exclusive. So again, it just kind of highlights how complex this whole system can be.
So in your book, in your pamphlet, you touched on mycorrhizae. Is that with potatoes when you're working with them? Is that the most common one you're using? Do you ever use bacillus strains and stuff like that? Or
Yeah. So we haven't been. We've done a little bit of testing on specific products. And this is something that's kind of interesting is like, so a lot of these products are like, they're tested in one part of the world or one, part of the country kind of thing, or on one species, and they're found to, say, promote plant growth. So like the bacillus that you've mentioned, or the specific mycorrhizae. But then you take them and you apply them to like the neighboring farm or on another crop and you don't see the effect. And there's a decent amount of literature that suggests that spiking a field with a single species usually doesn't have the effect that you would expect. And I think there's a few ways to explain that. And they kind of go to this kind of model here, right? So if your field, for example, you have, say, like high pH, and you add an acidifying bacteria, and that's, mobilizing the phosphorus, and you have, say, a phosphorus deficiency, right? That's going to have a much greater impact on promoting growth than if you did the same species into a more acidic soil.
So are any of these injected in mycorrhizae on any of these?
No, I'll get into what we're going on here. One of the things we're looking at is this idea of minimal input agriculture. And this, I'm kind of borrowing from the whole greenhouse gas carbon market, where there's the idea of equivalent CO2. So nitrous oxide that's released as a greenhouse gas is worth more CO2 molecules than, other species. And so we're kind of adopting that idea with the effective nitrogen. So one of the things we're asking is like, so if we have a field and we put, say, nitrogen in the form of nitrate versus ammonium versus, say, a beneficial microbe, how much does that promote plant growth? And so can we, for example, achieve a higher amount of plant growth or yield for a smaller amount of nitrogen, depending on the type of nitrogen it's found in. And that's where things like your mycorrhizae and that come in. So we haven't gotten to the point where we've started doing like inoculation trials. But I think the kind of first step is to sort of have conditions right where when you do introduce your beneficial microbe, it's able to take advantage of it. And this is where I think like things like carbon and that come in.
But I sell those bits in the boxes.
Yeah.
We're trying some big.
We haven't yet, but we're working up to it. I wouldn't mind speaking with you later on it. Yeah.
Couple things this is when it comes to nitrogen, so there's a cost to nitrogen, and so what's the trade-off for you and what you're saying?
Yeah.
So I guess one real there in front of them. The second part, there's background mycorrhizal in school. Background there because they adapt to the environment.
Yep.
And then we introduce perhaps less 100% adapted species. That's almost unique.
Yeah. A lot of interesting points on there. So when you're talking about the energy of nitrogen, are you talking about, say, the energy to produce the fertilizer, or are you talking about the energy that the plant expends?
So yeah, because the plant would have to expend the nitrogen energy to get its own nitrogen out of the air.
Yeah.
And not get it off. So
Yeah.
there's X amount of energy actually not kilojoule required per pound of animal.
Yeah.
So but if it comes from a resident bacteria in the soil,
Yeah.
then that's not affected by the plant.
Yeah, so plants themselves can't, yeah, so we're just discussing sort of the energy balance, and we're talking about nitrogen dynamics. So nitrogen, plants themselves can't fix nitrogen from N2 into nitrate. So that's where the bacteria is required, okay? Now, when the plant takes up the nitrogen, depending on what form it is, it needs to expend, well, it's not really, it's expending energy in order to assimilate that. So the plant needs the nitrogen to produce protein, essentially. And then it uses that protein as like building blocks for cell growth. And then if it's, a crop that accumulates protein, it's doing that as like a storage molecule in that. And it can break that nitrogen back down into energy. It's not as energy dense as, say, sugar. So I haven't studied this in terrestrial plants, but my previous life before I studied potato was on single-celled algae. And what we were finding was that, so the plant absorbs light energy, and we think about photosynthesis as it converts that into sugar with CO2. But it also uses the same cellular machinery in order to convert nitrates into ammoniums, which then get tagged onto for amino acids. So there is an energy cost. The question is, though, whether that's a cost that the plant can afford or not. So what we found with the single-celled algae is that it actually had a photoprotective effect. So when the plants were growing in excess light conditions, they actually have to expend effort, not necessarily energy, but effort in order to reduce the amount of damage caused by the light. Yeah, so we call it non-photochemical quenching. So photochemical quenching is that light energy gets tagged onto carbon and it makes sugar. Non-photochemical quenching is it basically reorganizes its cellular machinery and then gets rid of it as heat or fluorescence. If it actually could potentially promote growth if it's alleviating the negative effects. So it's not as clear cut. It really depends on the system and what's going on. But we also think about a lot of times as plants as being photoautotrophic, so photo is light, autotrophic is self-feeding. So most plants can, if they have to, generate all of their energy and their growth from inorganic sources. So they can take up CO2, they can take up nitrates and that kind of stuff and then grow that way. But if they have the opportunity to make use of a reduced carbon, they'll do it. And potato is an interesting example of that. So you can grow potatoes in complete darkness if you give it sucrose. Okay, so they're kind of opportunistic. If there's carbon in the soil that they can use to promote growth and they can take it up and use it, they will do that. And so this is where, say, like your microbial biomass kind of comes in, right? So it's more efficient for a plant to assimilate, say, free amino acids and use that directly than it is to convert nitrate into an amino acid. The thing is that everyone else is thinking the same thing, right? Things like iron, for example. Iron very quickly reacts with oxygen and then becomes rust, right? So microbes that are able to produce chemicals that keep that iron from oxidizing, we call them chelators. They're able to essentially increase the amount of iron that they're able to take up. So iron is in most systems is very limiting. Not because there isn't iron around, it's because it's an inaccessible form. You might have heard, like, you know, with the whole climate change and trying to capture carbon, the idea of fertilizing the oceans where they just kind of throw a boat out into the middle of the ocean, they throw iron into it. That's a big reason why it stimulates the microbial community, particularly plankton. So yeah, and again, I think this kind of gets to one of the major points is, like, you know, what you need to do to promote growth the best depends on the system you're working with. Does that answer the question? Kind of?
Good to say.
Okay, yeah. Okay. So again, all these functions are not mutually exclusive. That you can use multiple models to explain what's going on in a field. And I've got a very wet piece of paper, which is fine. Okay. No, that's fine. And so this kind of moves us into the next section on page 4, where the idea of assessing soil health. Do you want to get the next stuff up and we can show that? Okay, so general rule of thumb, again, not always, but general rule of thumb is that adopting practices to improve soil health will have positive benefits to plant health. Whether they're the benefits that you want depends on what's potentially wrong with your system and what you do to try and fix it. So this is why I think agronomists and growers, they know their fields best, they know their operations best. They're going to know what, you know, practices are going to best incorporate into their existing or which ones are potentially worth the effort. But we've already kind of talked about it where, like, there's all kinds of information out there and recommendations. I don't view my role to say, like, this is the best way to promote soil health because, you know, as we've already discussed, it's very complex and it's very contextualized. So a lot of what we're working on right now is kind of asking this question, well, are there better ways or ways to generate new types of data on soil health that could let us answer these questions or actually ask very different questions? So a lot of what we're focusing on isn't saying, like, carbon's the thing that you need to measure. It's asking if we could, say, measure carbon using a high density method or a high throughput method. So do thousands of samples instead of a dozen samples, for example, and generate, say, like a map of the field. If we could generate that type of data, how could it be used by an agronomist or a grower in order to make better decisions on how they manage their farm and address problems that they might have? There's lots of metrics that can be measured with soil health. And again, which ones are important depends on, you know, what issues you're facing and what problems you want to solve and what objectives you have, right? But there's a few of them that are pretty universal and or are easier to measure than others. And so what I'm going to talk about for the next bit here is 3 of them that we're looking at that kind of have that combination of they're pretty easy to use, they're universally or pretty much universally accepted as important things to measure. But keyly, there are things that we think we can either use automation or robotics in order to increase the density of sampling. So I've got a student, Marissa, wherever she is. We're gonna try and do a little bit of a hands-on thing. I don't know how well it's going to work, but she's gonna come around and show off a piece of equipment we're working with. This one is looking at total carbon, and we're on page 5. So when we are assessing total carbon, we're using a combustion method. So we take samples, we basically burn them, and then we measure the amount of CO2 that is produced. It does have the advantage of it also does total nitrogen at the same time, so you're getting 2 measurements at once. And there's other ways you can measure it, and depending on the method that you use, it's more or less labor intensive, and it also requires specialized equipment. Most testing facilities will be able to measure total carbon. What we're trying to do is see whether we can increase the amount of sampling that we can do for the same amount of effort, but also trying to do a non-destructive method to assess it. And so what we're using is, and then the other kind of objective is to try and use equipment that is easy to use and is relatively cheap. And again, it's like this idea, it's like if we put a tool in a grower's hand, what can they do with it? And then we're trying to figure out whether that's actually worth pursuing or not. And so cost of operation is a big barrier. So if we can reduce the cost of operation, that's a plus. So this is, it's called the NICS. It's made by, if you just Google NICS color spectrometer. But essentially it's a little electronic eye. And so what it will do is you put it up against whatever you're measuring, and then using your phone or a tablet, it will take a spectral measurement. Now, this is essentially the same technology when like you have run out of paint and you're painting your house and you've got to run out and grab a can and you want to try and have the same paint color. You take a little chip and they scan it. It's essentially the same thing, except we're using it to get the spectrum instead of trying to do a color match. So a couple example curves are shown there. The one in blue, the numbers next to it is the total percent carbon in that sample. So if we have one that's just a little over 1% carbon, and then another sample that is a 1.8. And the wavelengths are on the x-axis and the absorbance on the y-axis. And you can see where the 2 curves differentiate from each other. So in those wavelengths where we're seeing a difference between those 2 samples would be the informative part of the spectrum, okay? So what we're trying to do is see whether we can use those points on the curve in order to model the percent carbon that is present, particularly in samples that we haven't actually taken the measurement in yet. And so that's what's shown on the graph on the bottom. So the x-axis is that percent carbon, and then the prediction is based on the y-axis. So we're getting fairly good modeling between it. Now, the advantage of this is that, and you'll kind of see it as it goes, it takes just a few seconds to take a measurement. Now, we have to control for water because water is a confounding signal, so we're like doing an oven dry on this. But the other advantage is that we're using a very small amount of soil. So we're talking much less than a gram. You'll see it when it comes around. But because we're measuring it non-destructively, all the other measurements that we'll talk about, we can do on the exact same sample. So instead of taking an average measurement from one sampling method and an average measurement from another sampling method, we're actually taking multiple different types of data from the exact same sample. So why this is important is because it changes the types of statistics that we can use. So it would be analogous to, say, if we use a human health, we take measurements of Joseph's health, and then we try and determine what my health is. Right? So you're kind of taking a measurement of a population and trying to say, oh, well, you kind of look like this person in the population, you must be like that. Now, for a lot of things that holds true, right? Like if I'm overweight, I probably have blood pressure problems. Don't tell anyone else that I told you that. Whereas with this, it would be like taking that person's individual measurements and then saying this is what they are. So it allows kind of a different type of statistic. The next one we'll talk about, you can get the trays out for the slaking. The second one we're looking at on page 6. I don't have an example of this because it was really wet and this is wet and it wouldn't have worked out very well. But this is looking at water-soluble protein. So in molecular biology, it's a pretty standard technique to measure protein in samples on page 5 there. And so with this, what we have is we take the soil samples, we boil them, and that basically ruptures any bacterial and fungal cells in it. So the cells pop open, and then all the protein that's inside of them gets released and dissolved in the water. And then we take a small sample of that, we put it into a solution, and it'll change color depending on how much protein is present. So you can kind of see on there that there's different intensity blues. So the more blue it is, the more protein is present. So water-soluble protein is a quick proxy for microbial biomass estimation. So if you have more microbial cells, so more bacterial cells, more fungi cells in a sample, you would expect to see more protein. Now, it doesn't tell you how much of that biomass is living cells versus dead cells. To do that, we usually do a CO2 burst. So you take a dry soil sample, you wet it, and then you measure the amount of carbon dioxide coming out, and that's kind of an indication of how much of the microbial community is actually living and respiring. But it's a very quick method to kind of get a rough estimate. It's like, so do I have a lot of microbes in the soil or very few microbes? And the advantage of this is, again, like the sample we took with the spectral measurement, we could do this onto it. We also, I don't have results or a demonstration of it, but we're also looking at different fractions of nitrogen. So we kind of already talked about how it's different for the plant if it's given nitrogen in the form of nitrate versus ammonium and all that. So using this kind of approach where we're putting this onto a plate and then taking color measurements, we can essentially do different fractions. So we get total nitrogen from the combustion method, and hopefully we'll eventually be able to model it with the scanning. But then we can say, like, so X amount of it is in a nitrate form, X amount of it is in protein, X amount of it is in ammonium, etc. And so what we can ask is that within a particular field, how much nitrogen is present, but where is it actually in that whole system? So the nitrogen that's available for the plant is going to be in the ammonium phase. It's going to be in free amino acids. It's going to be in nitrates. Nitrogen that's in the microbial community protein is going to be locked in those cells, but eventually those cells are going to break down and release. So what we're trying to get a sense for is, like, how is the nitrogen moving through the system? And that's itself an indication of soil health, but it also lets us kind of get a sense for, so if we do this to the field, so we do a green manure, or we add cattle, like how much of that gets trapped and is like taken up by the plant? How much is the microbial community holding on to it? If we're doing greenhouse gas, we can get an estimation of how much is being lost. And you can also measure, like say in the water column, to see how much is leaching out. So you can kind of get a sense for, so when we do X particular intervention, where is the nitrogen flowing through? And again, we're trying to find ways to do it that are quick and easy so we can generate maps. You can go ahead and start handing that out.
Hey, Jonathan, how would you differentiate the microbes that are either beneficial to the plant or negative?
So that's somewhat difficult. There's a few ways you can do it. So essentially what you're trying to get as the identity and then assigning a function to that identity. So one way is you could extract DNA and then do a species or a genus level analysis, or even look for particular enzymes. You can go ahead and start handing that out. So there's been a lot of research kind of looking at nitrogen dynamics in terms of nitrification bacteria or nitrifying bacteria versus denitrifying bacteria. So some microbes or bacteria are going to be responsible for converting N2 gas into the different nitrogen species, so nitrates, nitrites, nitrous oxide, etc. And then each step can be performed by different species. So there has been some work done where they'll say extract DNA, they'll sequence it, and they'll ask like, so how many of this category do I have? How many of that category do I have? So that would probably be the way to do it. But that gives you, can give you an indication on pathogen load. It doesn't necessarily mean that if you have a high or low pathogen load, you're actually going to have the negative outcome or not. But it's a piece of the puzzle. Like I think, Michelle, like we've been seeing that with some of the verticillium, where like you assume based on other species that these are the verticilliums that cause early die, but then when we look in our soils, we see early die, but we don't find those ones. So is it caused by something else? And early die is an interesting thing too, because it's affected by, you know, the environmental conditions as well. So probably the fastest thing to do would be some kind of DNA sequencing or some other method to look for whatever function can be assigned to it. And there's some other ways I can talk about later. They'll need water. You can just, yeah. And then just a little bit. Okay, so this is the hands-on part of the presentation, or one of them. So another important measurement is aggregate stability. So commonly aggregate stability is measured using a wet sieving. So you take a soil sample, you put water through it, and then you see how much kind of comes out the other end. That's relatively time consuming. You have to use large samples. And so what we're working with, and again, we didn't develop this method. It's done by the Soil Health Institute. What we're trying to do is see whether we can scale it up so we can do hundreds of samples at a time. And so what we're calculating is something called a slaking index. So the students are coming around, they're going to give you, you've got a petri dish, they're going to put a bit of water into your petri dish, and then they're going to give you a piece of dried soil. And what you're going to do is you're going to hold the petri dish steady, and then you're going to drop the soil in, and you're going to watch mud be made.
The water should be water first.
Yeah, water first, yeah. And then,
Oh, yours is working.
so the basic idea here is that when the soil is dry, the microbes in it, they're producing mucilage and carbohydrates and stuff like that, and it essentially acts as a glue. And then when you rehydrate the soil, that dissolves these carbohydrates, and the soil will start to break apart. So as you drop it in and you watch it, it'll kind of soak up some soil, and then you'll see like parts of it fall off. Kind of like, you know, like soil erosion where it's all falling apart. So the rate at which it falls apart and the ratio of how large it gets when it's done falling apart versus how large it was when it started goes into this slaking index. So what we do with this is we take the dry soil, we spray water onto it, although you can do it the other way and drop it in. And then we take a picture every 10 seconds. And then we pull out of that picture where the soil is, and we calculate the change in area. And so an example on page 7 there, this is what the PEDs in this example look like when they're dry, and then after 10 seconds, so on. You'll see it kind of breaking apart as you look at it. And then what's shown on the graph is time on the x-axis, and then this slaking index on the y-axis. So you can see that initially the soils break down really, really fast, and then they break down slower, and eventually they reach a point where this is as large as they're going to get. It's a little small, but you can see the equation above the graph that describes this curve. And it's something called a Gompertz curve. So this is actually used quite a bit in disease modeling. So say COVID infections. So initially, when a virus first becomes present, it has a low infection rate, so a few people are getting it, but then it evolves over time, or more people get exposed, and suddenly your infection rate goes really high, and then people start getting immunity, and then it plateaus off. So each one of those components in that equation is part of the slaking index. So the rate at which it starts, the rate it initiates, and then where it ends up are the three components. And in our case, those are related to physical and chemical parts of the soil. So what we're caring about is this microbial mucilage, the glue that's holding it together. But things like your clay content, whether you've got cations in your soil, your pH will also affect it. So the absolute number isn't what matters so much. It's more about changes over time and when you do things. And we're going to talk about that in a second as a little example. But the thing that's nice about all three of these methods, there's a couple reasons why we're using these. One is that they're scalable. So the slowest step in all three of these things that we're doing right now is this slaking. The setup we have, we can do 40 pieces of soil, and we usually do 5 samples per core. So we can do, 8 cores in 10 minutes. And that's only a limitation based on, I set the camera to this height and this is how much I can see, right? Like you could have it on a conveyor and have it going a lot faster. I should say actually the slowest part is drying the soil, but once you got dry soil, it's pretty quick. The other components like the NIX is very quick, you saw that. And the automation on the robotics, the idea there is that you would measure multiple things. But typically that takes 30, 40 minutes.
The mix has to be in contact, essentially.
Yeah, yeah.
So are you doing the top 5 centimeters? What sort of depth are you looking at?
Great question. It depends on what you're trying to get at. So there's a trade-off between your depth of sampling. So what we're talking about is increasing the number of samples we can process per unit time. So within a day, I can do, say, 1,000 measurements. Now, whether that should be 1,000 cores in a single field, 100 cores for one field, and 100 cores for that field, or whether it should be like 200 where the first 100 are in the top 6 centimeters and then lower, depends on the question you're asking. So for our purposes at the moment, what we're trying to do, and I'll talk about it here in a second with the example, is we're trying to get a sense for what's the variation in the field. So it's more important to cover topographical, and so we decided we're just going with the top 6 inches, but we're going to sample, say, 100 cores, right? So it depends on what you're trying to look at. If you're trying to do, say, a carbon stock and like long-term carbon stock, then depth is going to be more important, and you might care about that top, right? So it all comes back to what you're trying to accomplish. But yeah, great question. Yeah, okay. So in the example, I'll just kind of quickly go through it here. In this example, the question we were kind of getting at on page 9 was, so if we went to, say, a really short rotation with potatoes, so a 2-year rotation, we know that's really bad for soil health. So every time you till the soil, you lose 50%, you disease, pressure, erosion, all of that. One of the questions we were trying to ask is like, well, so if in the intervening year we added a soil health promoting practice, could we offset some of the negative? Okay, so that's what we looked at. I'm only going to show the slaking information, but on figure 6 is showing that slaking index. So higher numbers are worse. You want lower numbers. In this example, potatoes were growing in 2020. And then in 2021, we took a sample in spring and fall. And in that year, we grew a green manure crop with cattle grazing. So what you can see is the slaking index started high after the potato, but then when the green manure was included, that slaking index decreased. So this is indicating our soil aggregate stability is increasing, and we expect that other parameters are going to improve as well. But the next year, we went back to potato. And what you can see is that the number went right back up. So a couple points with this. One, using these methods, we're able to sort of show very short-term changes. So these methods are really good at capturing an instant time point. And because they're relatively easy to measure, you could say measure it every month. Like what the limitation there would really be how many people can you get out. And if you've got like an automatic core that's kind of going around like that, it becomes pretty easy. Yep.
Timing of sampling, because you're talking about, okay, you spread it for 2022, the numbers were high, what year was that? After the potatoes were planted?
It was, so the seed bed prep was in fall after we took the fall 21. So this would be before the potatoes were planted, but the seed bed was prepped. And so we think that tilling of the seed bed is probably what's accounting for a big difference. Yeah. Now, that number right there is looking at the average for the entire field. What I want to point out, and where I think these approaches are really useful, is looking at, you know, is the field homogeneous? So that's what's shown on the remaining figures here. So we're looking at spring 21, spring or fall 21, and so what you can see immediately is that the field is not equally homogeneous across the entire field. There's pockets where the soil health is better or worse. But one thing that is interesting is when we do some math to compare the two time points, which is the last figure, so this is the difference between them. So what you see is that overall the field improved, but there was actually a couple pockets where the soil health parameters actually got worse. Okay, so if we had done like a pooled sample for like a single point in the field, you wouldn't be able to calculate these contour maps. And I think that's the advantage of these approaches. I wouldn't advocate doing this in lieu of what you're currently doing. The idea is, does this give you actionable information beyond what you're currently doing? And is it doing it at an economically beneficial, and how do we make use of that? And so that's, I'm running out of time, but that's more or less what we're trying to do with our research.
Thank you very much, Jonathan. I will give you this.